Hello students, welcome to EduRev. Have you ever wondered where life first began? And how tiny building blocks called cells made everything possible on Earth? Today we will explore a gentle journey from boiling hot springs on ancient Earth to the busy inner life of a single cell. And by the end, you will recognize the key parts that make living things work. Imagine small pools of warm water on early Earth, like the hot springs of Puga Valley in Ladakh, where heat-loving bacteria called thermophiles tucked themselves into calcium carbonate deposits and found shelter from harsh conditions. Those mineral coatings may have protected early organic molecules and helped form the very first membranes that define a cell. And that idea helps us picture how life could begin in tiny, changing environments rather than in a vast ocean. Now let's bring that picture down to the level of something you can study in class. A cell is the basic structural and functional unit of all living organisms, from a single bacterium to a giant tree or a human being. Some organisms are unicellular, made of just one cell like bacteria and amoeba, while others are multicellular, made of many specialized cells that form tissues, organs, and organ systems working together. Think of a multicellular organism as a well-organized team. Cells become tissues, tissues form organs, and organs join into systems like the respiratory system that helps you breathe. Cells are tiny and cannot be seen with the naked eye because the human eye cannot resolve objects closer than about 0.1 millimeter. So scientists use microscopes to reveal their hidden world. Robert Hooke was the first to observe cells in 1665, when he looked at cork with a microscope and noticed small box-like compartments that he called cells. And his discovery opened a window into life's inner architecture. There are two main kinds of microscopes you should know: the light microscope used in school labs, and the electron microscope that uses electrons to show structures at the nanometer scale. Over time, three features improved and changed how we study cells: resolution, which controls clarity; contrast, which shows differences in brightness; and magnification, which makes things look larger. A simple classroom activity helps estimate cell size by dividing the diameter of the visible field by the number of cells across it, and remembering unit conversions like 1 millimeter equals 1000 micrometers. Every cell has a boundary called the cell membrane that surrounds and protects it and controls what passes in and out, which keeps the cell's internal life balanced. The cell membrane follows the fluid mosaic model, a thin lipid bilayer with proteins embedded like gatekeepers, allowing movement of molecules while remaining flexible like a living mosaic. Particles move naturally from higher to lower concentration by diffusion, and water moves through a selectively permeable membrane by osmosis, from dilute to concentrated solutions until balance is reached. Solutions affect cells differently. Isotonic solutions leave cells unchanged. Hypotonic solutions make cells swell as water enters, and hypertonic solutions make cells shrink as water leaves. You can see osmosis in action with the potato experiment. Potato pieces swell in plain water and shrink in a concentrated salt solution, showing how water flow changes cell volume. Plants have an extra layer called the cell wall, made mostly of cellulose, which gives rigidity and keeps plant parts upright even when the inner membrane pulls away during plasmolysis. Animal cells lack a cell wall and can change shape more freely, which helps tissues move and perform different functions. Inside each cell, there are three basic parts: the cell membrane, the cytoplasm, which is a jelly-like fluid, and the nucleus, which acts as the control center. Eukaryotic cells contain many membrane-bound organelles that specialize in specific jobs, while prokaryotic cells are simpler, lacking a true nucleus and most organelles and often having DNA in a nucleoid region. The nucleus holds DNA and genes, and it is surrounded by a double nuclear membrane with pores that let information pass to the cytoplasm, while the nucleolus inside helps build ribosomes. Ribosomes are tiny protein factories that read DNA instructions and assemble proteins, and they can be free in the cytoplasm or attached to the rough endoplasmic reticulum. The endoplasmic reticulum is a network for making and transporting molecules. The rough ER has ribosomes and makes proteins, while the smooth ER makes fats and hormones. The Golgi apparatus acts like a post office, modifying, sorting, and packaging proteins and lipids into vesicles for delivery or secretion. Lysosomes are the cell's cleanup crew, filled with digestive enzymes that break down waste and damaged parts so their building blocks can be reused. Mitochondria are the powerhouses of the cell, where cellular respiration produces ATP, the energy currency that powers most activities. And interestingly, they have their own DNA suggesting an ancient origin. In plant cells, plastids such as chloroplasts capture light for photosynthesis and store food. And like mitochondria, they contain their own DNA and specialized internal structures. Vacuoles store water and nutrients. And in plant cells, the large central vacuole keeps the plant firm by maintaining internal pressure called turgor. Cells grow and divide to replace damaged tissues and to help organisms grow. And we do this through controlled processes like mitosis and meiosis. Mitosis produces two genetically identical daughter cells for growth and repair, while meiosis produces four genetically varied cells with half the chromosomes for sexual reproduction. Cell division is carefully regulated because errors can cause tumors or genetic disorders, and normal cells often stop dividing when they touch neighbors, a control called contact inhibition. Some cells are programmed to die through regulated processes called programmed cell death, which is essential for development, like shaping fingers by removing the cells between digits. The classical cell theory sums up these ideas. All living organisms are made of cells. The cell is the basic unit of structure and function, and all cells arise from pre-existing cells. Scientists like Hooke, Schleiden, Schwann, Virchow, Golgi, and later researchers refined these ideas and gave us the tools and concepts we use today to study life. If you pause and reflect, the main insight is simple but powerful. A single cell contains organized systems that together sustain life. And understanding these systems helps us explain growth, health, and disease. Quickly recapping. Life likely began in small watery habitats. Cells are the fundamental units. Membranes control exchange. Organelles perform specialized jobs. And cell division and death maintain life's balance. Remembering these basics will make complex topics like genetics, physiology, and biotechnology easier to follow as you go deeper into biology. Thank you for listening gently and staying curious. And if you'd like more animations, practice questions, or experiments to explore cells, come learn with us on EduRev. Stay kind to your curiosity. Keep asking small questions that lead to big answers. And we'll meet again for the next lesson on EduRev.